Hello, and welcome to this lecture in which we'll look at embryo folding and development of the embryonic cavities and the placenta. Now, I should say at the beginning that this is quite difficult to understand. It probably took me several years to get my head around it, and you may have to watch it several times before it makes complete sense for you. And what we're going to do is to look at, first of all, the folding of the embryo, and then how the extra embryonic cavities and membranes develop, and then finally we'll look at the development and the function of the placenta. But we have to begin right back at the very beginning, at the point of implantation. And in this diagram, we can just see the pre-embryo beginning to implant under the uterine wall. And the outer cells, the trophoblast, as we described in an earlier lecture, actually begins to form syncytiotrophoblast, which has lost its cellular boundaries, and you have nuclei in a space surrounded by cytoplasm. And this is eating its way into the uterine wall. And we can see that the primitive ectoderm, here marked in red, and primitive endoderm, marked in yellow, have already formed from the inner cell mass. The uterine wall has been prepared to receive this, otherwise, as we said previously, this would be quite dangerous as it implanted into the surface. 2 days later, it's almost completely disappeared under the surface, and primitive ectoderm, marked in red here, has formed a cavity, and that's going to be the amniotic cavity. That's kind of the thing to focus on. If you follow the amniotic cavity all the way through, that's probably the most important bit. The other ones tend to come and go, but the amniotic cavity is going to be really important later on. So at this point, it's sunk completely under the surface. Blood vessels are being engulfed by the syncytiotrophoblast, and maternal blood is in direct contact with the syncytiotrophoblast. So nutrients are beginning to make their way to the developing pre-embryo. As we said, the inner cell mass has given rise to the amniotic cavity, and just where the embryo is sunk under the surface, there's a little plug or scar which marks where it is. Quite hard to see, even if you're looking inside the uterus at this point. A little later still, 11 days after fertilization, now sunk completely under the surface. The amniotic cavity is still visible. The yolk sac is formed from the primitive endoderm, and we can begin to see little villi, little projections from the trophoblast, marked in dark brown here, projecting into the syncytiotrophoblast. And these villi are increasing the surface area of the developing conceptus so that nutrients can flow from the mother. So growth from now on will be relatively rapid. The primitive endoderm has formed that primitive yolk sac. And the primary stem villi are forming to increase the absorptive area. So rapid growth ensuing as a result of nutrients coming from the mother. In this image, we can see the yolk sac separated from the rest of the embryo with the amniotic cavity shown in green up above it. That's the amniotic cavity. As I said, that's the key part. The body stalk is what's going to be the umbilical cord later on. And the embryo itself is going to develop in the bilaminar disc. That's the part made up of ectoderm and endoderm in between the amniotic cavity and the yolk sac. As the cavity is expanding, it's beginning to push its way back into the cavity of the uterus, the uterine cavity overall. To remind you that when we talked about gastrulation, we saw the primitive streak forming in the floor of the amniotic cavity, and that defines the main body axis. Also gives rise to the mesoderm of the embryo, so that 2 layers of the ectoderm and the endoderm between the amniotic cavity and the yolk sac become 3 layers, with mesoderm as the meat in the sandwich, as it were. Now, if we were to follow the embryo itself, it begins to fold, and looking at it from the side, what happens is that as the body begins to curve into a C shape, so the whole embryo begins to rise into the amniotic cavity. And the amniotic cavity is swinging round front and back, and also, as we'll see, from side to side, so that gradually the embryo is going to be suspended, as it were, in the amniotic fluid, which fills the amniotic cavity. So that's the view seen from the side. And if we looked at a solid view of the embryo, you can see the same structures as it curves round to form this C shape. And at this point, you can begin to get the idea that this might be a vertebrate embryo, might even begin to guess that it might be like a human embryo, might be looking a bit like a human embryo from now on. Now we're going to see the same thing seen in transverse section, a cross section across the long axis of the embryo. Again, we've marked the amniotic cavity, and you can see how it's sweeping round from the sides as well. So the embryo rising up as the amniotic cavity filled with its fluid begins to sweep round the embryo and pinch off the yolk sac underneath. A little later still, that sweeping round process is continued, and here we can see how the embryo is rising up and projecting into the amniotic cavity, and the yolk sac is contributed towards formation of the gut. Now let's look at essentially the same process again, but we're going to step back a level. And now we're looking at it in the context of the whole of the cavity of the chorionic cavity, which is the large space that you can see here. You can see the amniotic cavity is marked again, the body of the embryo and the yolk sac. And what happens is that mesoderm will cover all of that internal and external surfaces, so covering over the amniotic cavity, covering over the yolk sac, and lining the extra embryonic chorionic cavity. On the right-hand side of the picture, you can see our amniotic cavity again, and again, I emphasize that that's the thing to focus on. That's expanding as the embryo folds up into the amniotic cavity, and the yolk sac will steadily diminish in size and relative importance. You can see on the outside how the trophoblast cells, the villi have become more complicated, and they're beginning to develop a branched structure as they continue to increase their absorptive area. And of course, that is surrounded by syncytial trophoblast, with maternal blood running through these spaces in the syncytial trophoblast. Little later still, again, focus on the amniotic cavity. And you can see as you move from 3.5 weeks up to about 5.5 weeks, that amniotic cavity is expanding, and gradually the chorionic cavity is diminishing in size. So it will go on expanding in that way. And the section view through the embryo you can see here shows the body of the embryo as it rises up into the amniotic cavity. Now, let's move from a diagram to some actual specimens. So here's a human embryo at roughly the same kind of stage of development. And here you're looking at the chorionic cavity. You can see the embryo inside its embryonic sac, so it's lying inside the amniotic cavity. And the little structure just to the side of the embryo is the diminishing yolk sac. Then surrounding all around the outside are the fronds of the villi, which are in contact with syncytial trophoblast and through which maternal blood is flowing. And you can already see that perhaps they're a little thicker towards the top of the picture than they are down at the bottom of the picture. And what's happening is the villi are beginning to become concentrated in one particular area. So looking at that in diagrammatic view, now you can see that the amniotic cavity is almost completely excluded all of the previous chorionic cavities, and the embryo is floating freely within that. And it's during the embryonic period that the embryo first becomes capable of moving. So it will show spontaneous movements, although these are still too small and gentle for the mother to be aware of them. But you can see how the villi are concentrating at one side of the external structure. Now let's step back another layer and we'll look at it in the context of the uterus. So here we're looking at a view, a section through the mother from the side, looking at the uterus. And you can see how the whole developing structure is beginning to project into the uterine cavity. And as the amniotic cavity gets larger and larger, obliterates all the other spaces, gradually it will come to almost completely fill the uterus. So at later stages, amniotic cavities almost completely occupying all of that space, projecting into the uterine cavity until finally it's almost completely excluded. All the other spaces are excluded by this amniotic cavity developing on the outside. The structures which are derived from the mother remain as called the chorion. So these are described as the chorion. So the amnion is meeting the chorion and forming a fused membrane once all the space is eliminated. The area where the villi is concentrated is called the chorion frondosum, and the remaining smooth part is called the chorion levi. And you can see in this late picture, 5 months into the pregnancy, that all of the space is taken up by the amniotic cavity, the amniotic fluid, and the baby within that. So for the baby to get out as it exits down through the cervix, then what's going to happen is it has to rupture those membranes that lie between it and the outside world. And of course, these chorionic villi, you can sample them. The ones which are going to disappear are obviously not important for survival. So you can insert a needle and take a sample of the chorionic villi and then analyze them for genetic defects. And that's called chorionic villus sampling. It is in itself a slightly risky procedure and therefore slightly safer to take a little piece of the amniotic fluid. So again, you can insert a needle in, draw off some amniotic fluid, not only analyze the biochemical composition of the amniotic fluid, but you can also look for embryo cells that have been shed into the amniotic fluid. And these can be cultured and again examined for genetic defects. Now, the things that we've been looking at are obviously very different to if you have a twin pregnancy, as we discussed in another lecture. And here we can see some arrangements of different versions of the membranes in the light of what we've been saying about the development of these species. So you can see on the left-hand side, if there's early separation into 2 separate blastocysts, then each of those will have separate membranes. So a third of the twins that we get are identical. And of course, that number, as we said previously, depends on in vitro fertilization, which can increase the number of non-identical twin pregnancies. But ones which are formed very early on may well actually fuse together. And you can see that in the form of conjoined twins. But we need to be able to say a bit more about the membranes themselves. So you can see in the image labeled A that completely separate development has taken place. There are 2 amniotic cavities and 2 chorionic cavities. So you would describe that as a diamniotic, dichorionic pregnancy. In the image marked B, by contrast, there are still 2 amniotic cavities, but only 1 chorionic cavity. So that would be a diamniotic monochorionic cavity. In this image, C is the same as the previous one, that's diamniotic monochorionic. But you can also see that you can have twins developing inside a single amniotic cavity. So that's a monoamniotic cavity. And finally, conjoined twins are within the same amniotic cavity completely. Now let's look at the placenta itself. And we saw in the earlier diagrams how the villi are becoming more complex. So this is a view in time with a time dimension along the axis, and it shows the increasing complexity of the villi themselves, projecting into the syncytial trophoblast with maternal blood flowing through the syncytial trophoblast. Eventually, embryonic blood vessels will develop inside the villi, but you can see that embryonic blood and maternal blood never actually come in direct contact with each other. Always have to cross a membrane composed of the trophoblast cells and the syncytial trophoblast cells in order to exchange nutrients and gases between the 2 of them. And again, here's a picture which we've looked at previously, which just shows you what that illustration might look like in real life. The diagram is very helpful from the point of view of labeling, but the photograph gives you an idea of what it would actually look like. Now, what's the placenta doing? And there are 5 major functions which are being fulfilled by the placenta, some of which we've mentioned already. So one of its major purposes is gas exchange. So oxygen is coming from the mother and being taken up by the embryonic and fetal blood. And that requires that that embryonic and fetal blood has to have a higher affinity for oxygen than normal adult human blood, so that the developing embryo or fetus can extract oxygen from the maternal blood across that membrane that we discussed previously. Equally, waste gases will diffuse back across there and into the maternal blood and be dealt with by the mother's respiratory system after that. It's not just gases that are involved, there are also nutrients and waste products. So nutrients being supplied by the mother to the developing embryo and fetus, and waste products being removed from the embryo and fetus into the mother's blood and then dealt with by the mother's own systems subsequently. And of course, if there were to be some adverse environmental factor present, then it may be able to pass the placenta. We know, for instance, that smoking will, during the course of pregnancy, will significantly decrease the size of the placenta and make it much more likely that things will go wrong during the course of the pregnancy. However, that's not the only functions fulfilled by the placenta. It's also a major site of hormonal action right from very early times, so that one of the early signals that pregnancy has occurred is release of human chorionic gonadotrophin into the maternal blood, and that's the basis of many pregnancy tests. That's mediated by the tissues that will become placenta later on. And then finally, the embryo is itself different. It's genetically different from the mother. So why is it not rejected in the way that the mother would reject an organ transplant unless she was given drugs to suppress her immune system? What is it that protects the embryo and fetus from being rejected as foreign tissue? And the answer is that we still have much to learn about this. But there are aspects of it that we know that are mediated by the placenta itself. So it seems to create an immunologically privileged site which allows the developing embryo and fetus not to be rejected. And of course, if we knew how that worked, that might give us clues for dampening down rejection of tissues in organ transplants. And therefore, that's a major site of research interest at the moment. This is a picture of a placenta seen after birth. The baby is born first and then placenta is delivered afterwards, as I'm sure you know. This is a slightly unusual one in that the umbilical cord is at the edge, but that's not a variant that would cause any difficulty or harm during the pregnancy. So what have we looked at in this particular lecture? We've looked at folding of the embryo. We've looked at the development of the extra-embryonic cavities, focusing on the amniotic cavity and the membranes that surround them. And then we closed by looking at the development and the functions carried out by the placenta. Thank you very much.